Dear students, in today's lecture we will discuss about cell division in eukaryotes. Like the process of reproduction in prokaryotes, eukaryotic cells reproduction also requires various processes that includes the DNA replication, then the separation of that replicated copy, and at the end division of the cytoplasm. However, the presence of multiple DNA molecules requires a more complex mechanism, so that each chromosome is replicated in a very well defined or in a very accurate manner. Here, this more complex mechanism is required because these chromosomes are separated by the nuclear envelope. As in case of the prokaryotic cells, that chromosome was present without any separation because of absence of membrane-bound organelles, or because of the absence of the cell membrane. But here, chromosomes are separated by nuclear envelope, and the nucleus was once thought to be fluid-filled bag in which the chromosomes are floated, but now we can see that nucleus is a highly organized internal scaffolding which is known as the nuclear matrix. And we all also know that there are two sets of chromosomes which are present in the eukaryotes. The presence of two sets of the chromosome is a consequence of sexual reproduction, because these two sets of chromosome, one set of chromosome is inherited from the male parent, while the other set of the chromosome is inherited from the female parent. And there is also diploid versus haploid pairs. Human cells, for example, have 46 chromosomes that constituting 23 homologous pairs. The two chromosomes of homologous pairs are usually alike in structure and size, and carries genetic information as same as homologous pairs are usually alike in structure and size, and carries genetic information for the same set of the hereditary characters. And sex chromosomes are an exception in this case. They are very different from the other chromosomes. So, first of all, if we want to understand in detail about the structure of the eukaryotes, we have to understand the structure of the chromosome. Eukaryotic chromosomes are basically very highly folded and condensed, and that folding and condensation is because if it is stretched out, some human chromosomes would be several centimeters long, thousands of times as long as the span of the typical nucleus. To package such a tremendous length of DNA into small volume, each DNA molecule is coiled again and again and tightly packed around the histone proteins. As we are aware that DNA in eukaryotic chromosome is coiled with the histone protein, which forms a rod-shaped chromosome-like structure. A functional chromosome has basically three essential elements. As you can see here, this is the functional chromosome, and it has three elements. First is the centromere, second one is the telomere, and third one is the origin of replication. Centromere, a pair of telomeres, and an origin of replication. So, first of all, we will discuss about the centromeres. Centromere is basically the attachment point of for the spindle microtubules. Is attachment point, the filaments which is responsible for moving the chromosomes in cell division. These spindle microtubules are basically the filaments which is responsible for moving the chromosome in cell division. The centromere appears as a constructed region before cell division, multi-protein complex which is called as the kinetochore that assembles on the centromere. So chromosomes which lack centromeres cannot be drawn into the newly formed nuclei. The chromosomes are lost, often with the catastrophic consequences for the cell. So on the basis of centromere, chromosomes are classified into various classes. First classification is the metacentric, and in this case you can see that centromere is present with exactly in the center, and in this case the length of the telomeres is same on both sides of the metacentric. While second class is the submetacentric. In this case, one of the two arms of the chromosomes is short of the submetacentric, or next is known as the acrocentric. Submetacentric, here one arm is shorter than the other arms. Third one is the acrocentric. In this case is also the one arm is very short compared to another arm, while in telocentric, centromere is present at very end of the telomeres. Next is the telomeres. Telomeres is basically the natural ends and tips of the whole linear chromosomes, and their function is basically the protection and stabilizing the chromosome ends. If a chromosome breaks, producing new ends, the chromosome is degraded at the newly broken ends, and telomeres basically provides the stability. Research also showed that telomeres also participate in limiting the cell division and may play important role in aging and cancer. Next is the origin of replication. This is basically the site where DNA synthesis begins. So these are the three basic structures within the chromosome. Thank you.